Beyond the Scope webinar. Today, it's going to actually be our last webinar of the year. So Carley's going to finish us up with talking about the microCT. We'll be picking back up again in January. The schedule's not quite finalized yet, but we should be finalized that within the next week or so. So we'll have a lot of different new topics, and we'll be keeping the same schedule of having them every other week. The time might be a little bit different. If you have any strong opinions about that, let me know, and we can definitely try and work on that schedule as well. So as with all these webinars, we try and keep them generally on the shorter side of things, around 25 minutes or so that you're able to ask lots of questions at the end. So if you have any questions during the webinars, you can ask those in the Q&A and chat. We do record this during the entire thing. It's available afterwards as well on our web page. So today, there's going to be Carley Goodwin giving a talk about the HeliScan microCT and some interesting information about some different scanning techniques. Carley.
Thanks for the introduction, Dan. And thanks also to you and Ashley Albertson for organising and hosting the discussion series this year. Good afternoon, everyone. Thank you for taking the time to join me today. My talk is titled Introduction to the HeliScan microCT and Region of Interest Scanning. I am Carley Goodwin, and I'm a research associate here at the Centre for Electron Microscopy and Analysis, and I manage our HeliScan microCT system. Let's quickly go through the outline for today's webinar. If you have attended any of the previous webinars, you will know we usually start with a couple of introductory slides, which give a bit of a background information about the presenter and also information about the instruments and resources available at CMAS. This will be followed by an overview of the HeliScan microCT, then the region of interest workflow. I would also like to mention that I've targeted this webinar at a beginner introductory level. I aim to keep this talk short, so there will be lots of time for questions at the end. Please place any questions in the Q&A box. If you have a longer question, there is also a raise your hand option, which you can use at the end of the session. Or if you have a question that you would prefer to discuss offline, please drop me an email and we can set up a Zoom call later. If you have any technical issues, please post a comment in the chat window and our moderators will try to help you. So a bit about me. I grew up in South Australia, and at the bottom of this slide, I've included 2 images from West Lakes, one of the places I like to go kayaking when I'm back home. I completed my Bachelor of Engineering at the University of Adelaide. After completing university, I moved to Canberra to work at Digital Core, a company which specialised in digital rock analysis. And this is when I first started working with the HeliScan. Digital Core went through a merger and became Lithicon. Then Lithicon was bought by FEI, who was later bought by Thermo Fisher Scientific. Then I spent a year working as a technical officer at the Tyree X-ray lab at the University of New South Wales before joining the team at CMAS in 2018. CMAS is a custom-built electron microscopy centre, and we have a large range of electron microscopes and other analytical techniques. This includes our Titan and Themis TEMs, and on the cryo side of TMs, we have a Glacios and Krios. We also have 2 focused ion beams and 4 SEMs. We also have an XRD and nano indentation capabilities. And today we are going to be talking about the HeliScan microCT. So what CMAS resources are available? We host the Beyond the Scope CMAS Discussion Series, which you're attending today. We also offer one-on-one consultation. If you're interested in learning more about any of the instruments at CMAS or discussing a project, please reach out to one of us. We also offer limited remote connection to our data analysis computers to analyze the data you collect at CMAS. So now let's talk about microCT. Taking an image using a microCT is similar to if you're going to go to the hospital to get an X-ray of your chest or your arm. If you're getting an X-ray, you'd be in between an X-ray source and a detector, just like the sample is in this image. The sample in this image is a chocolate Kinder Surprise egg. The X-rays would pass through you and be attenuated by different amounts depending on the material they are passing through, and the detector would capture an image. With a microCT, instead of just taking the one image, the sample is moved through a set trajectory and a large number of images are captured. These images are then reconstructed and turned into a 3D tomogram. MicroCT is a powerful research technique as it allows us to non-destructively capture 3D information about our samples, and it can be used to investigate internal and surface features. Samples can also be scanned before and after, or at multiple stages during an experiment, and MicroCT can be incorporated as part of a multi-scale correlative workflow. What are some of the benefits and advantages of using the HeliScan microCT at CMAS? The HeliScan can perform both circular and helical trajectories. It has a vertical Z stage, which can move 195 millimeters vertically. This combined with the helical scanning trajectory means that the samples with large aspect ratios can be scanned in one image. The sample stage can hold a maximum of 15 kilos, allowing for heavy samples or cells to be installed. The HeliScan has two scanning modes, the first one being the high resolution low flux mode using a Lab 6 filament. The smallest achievable voxel size in this mode is 400 nanometers, and the maximum tube voltage that can be used is 100 kV. This mode is good if you want to image a smaller sample that is low attenuating. The second mode, which the system can be operated in, is the lower resolution high flux mode using a tungsten filament. The smallest achievable voxel size in this mode is 800 nanometers, and the maximum tube voltage that can be used is 160 kV, making this mode good if you want to image larger, more dense samples. Achieving these small voxel size is dependent on your sample geometry, and you need a very small sample, say about 1 to 2 millimeters in diameter to achieve them. Or if you have a slightly larger sample, you can use region of interest scanning. The HeliScan has a region of interest workflow, which we'll discuss a bit later, and it also has a multi-scan workflow. The multi-scan workflow allows you to either take images of the same sample at different scan settings, or to stack several samples on top of each other and set up individual scans for each one. This is a great option if you want to run a few samples over the weekend. And there's also phase contrast retrieval, which is useful for low attenuating samples. The HeliScan at CMAS has a range of users from across the OSU campus, including the ones listed on this slide. We also work with researchers from other universities and industry partners. On this slide, I just wanted to showcase some images from our users. Samples A, B, D, and E and F are 2D slices from tomograms. You can see a bee's head, a slice from some titanium 6-4 powder, a slice from an additive manufactured sample, a piece of food biscuit which has been compressed, and a piece of sandstone. The mouse heart in image C shows a 3D volume rendering. The pulmonary valve is highlighted in green. The rest of the mouse heart is the gray-white color, and the resin block it's mounted in is the brown color. Here you can see the overview image of the workflow for the scanning a sample on the HeliScan. You take your sample, scan it, acquire a set of projections, reconstruct them, and a tomogram is produced. This first part of the workflow is performed on the HeliScan, with the final data set being produced being a tomogram, which is exported in either a TIFF stack or a set of net CDF blocks. Once you have your tomogram slices, they can be loaded into an image analysis program for visualization and analysis. Over the next few slides, we're going to discuss all these points in more detail. So the first step if you want to scan a sample on the HeliScan is going to be to get a sample and then mount it. For this webinar, I'm going to use this sweet gum seed as my sample. So now I have my sample, I need to mount it. Since this sample is an irregular shape, I just use some Play-Doh to create a more flat surface on the bottom, and then use some double-sided tape to attach it to a sample stub. When mounting a sample for microCT imaging, it is important that your sample will not move during the scan. Any sample movement could cause any significant sample movement during your scan will result in a bad reconstruction. The first type of scan we're going to talk about today is a full field scan. As the name suggests, the aim of a full field scan is to optimize the amount of the detector being used, but not let any of the sample go out of the field of view. There should be a small amount of air along the vertical edges of the images captured by the detector. This is the standard scan done on a microCT. Now back to our sample. Once the sample is mounted, it can be placed in the microCT. In this photo, you can see our sample, the X-ray source, the sample stage, and the detector. When setting this scan on the HeliScan, I used a sample distance of 38 millimeters and a detector length of 330 millimeters. I also added a filter to the detector, and that's a silver plate you can see just at the arrow where it says detector with aluminium filter. I also chose to use a space-filling helical trajectory, and I set the exposure time. And I also set all other required parameters. So this full field scan ended up taking around 2.5 hours. When starting a full field scan on the HeliScan, the sample size will limit the smallest achievable voxel size possible. This table shows what the approximate voxel size attainable for different sample diameters are. For example, if you have a 5 millimeter diameter sample mounted on a post that is either 5 millimeters or smaller, so it doesn't add any extra diameter to your sample, you could get a voxel size of around 2 micron. However, if you have that 5 mil diameter sample, but it's in a tube with a wall thickness of 3 millimeters, the sample is effectively 8 millimeters in diameter, which limits the voxel size you can get. So in this case, you can get a voxel size of around 3.1 micron. For the sweet gum sample, we have a sample just under 40 millimeters. So I was able to get a voxel size of about I was able to get a voxel size for the tomogram of 16 micron. I may have been able to get a slightly smaller voxel size. However, not by much. If I'd optimized the settings a bit more, maybe it could have been slightly more like 15.8 or something, but it was pretty much the best I could get. Once the scan settings have been set, the acquisition is started. A series of projections will be collected as the sample is moved throughout its scanning trajectories. This is a small subset of 16 out of the 2,861 projections acquired during the scan of the sweet gum seed. Clear fields, projections with X-rays switched on and no sample in the field of view. Dark fields, projections with the X-rays switched off. And keyframes, which are small subsamples of projections which are retaken at the end of the experiment, will also be required. Once the scan has finished, all of that acquisition data will be transferred from the acquisition computer to the reconstruction cluster, where the data will be reconstructed using the reconstruction algorithms. After reconstruction is completed, the final output for the HeliScan will be a series of TIFFs will be a tomogram exported as either a stack of TIFFs or net CDF blocks. On this slide, you can see 3 slices, so Z slice 500, Z slice 1000, and Z slice 1500 from the tomogram of the sweet gum. Then you can take your reconstructed slices and visualize and analyze them in an image analysis program. This image shows the data loaded into a VISO. You can see the X, Y, and Z central slices in 2D, and you can also see a volume rendering of the data in the top right-hand corner. CMAS users can also get access to our virtual machines to analyze their data. We currently have both a VISO and MyPar installed on that virtual machine. There are also other image analysis softwares available like Dragonfly, TomVis, and Slicer 3D, and there's also many more available. Now that we have our reconstructed tomogram of our sample with a voxel size of 16 micron, what happens if there are features we can't resolve? Or if we just want to explore the sample and see which features, see what we can see if we had a smaller voxel size. In this case, we have 2 options using the HeliScan. We can either physically cut the sample, so we'll make it into a smaller sample with a smaller diameter, and then we could perform another full field scan. This is okay if you don't mind your sample being cut. However, if for some reason you don't want to cut into your sample because you need to use it for other experiments, or you just don't want to destroy your sample, you could do a region of interest scan. This will mean that we increase the magnification and let some of the sample go out of the field of view. When we set up the region of interest scan, we are going to increase the magnification of the sample onto the detector by moving the sample as close as physically possible to the source and moving the detector as far away as possible. Unlike in the full field scan, we will let a large amount of the sample go out of the field of view. And on this slide, you can see the difference between a projection from the full field scan and a projection from the region of interest scan. When we're doing a region of interest scan, the final reconstructed volume of the region of interest scan will only show the sample selected for the scan. However, the X-rays which are passing through that region of interest are also passing through your sample. So they're not just being attenuated by the part of the sample that you're reconstructing, they're also being attenuated by material which you are not reconstructing. The HeliScan has a 2-scan region of interest workflow, which allows the users to take a fast, low-resolution scan of the whole sample, as well as a longer, high-resolution region of interest scan. The reconstruction of the overview scan is used. The reconstruction of the overview scan is used by the reconstruction algorithm when they're reconstructing the region of interest volume. This way, the reconstruction takes into account that the X-rays that pass through the region of interest also pass through other parts of the sample which aren't part of the region of interest area. The scan has to be imaged with the same source settings as the region of interest scan, but the resolution and quality don't have to be the same. So for the overview scan, I used a detector binning, which increased the voxel size and also decreases the scan time. When setting up the region of interest scan, the settings are optimized to capture the maximum possible resolution. Only the features captured in the projections will be reconstructed. Any other features will not appear in the final reconstructed dataset. The image on the left-hand side shows a 2D slice from the overview scan. The red circle shows approximately where the region of interest scan was taken. On the right-hand side, you can see a slice from the reconstructed ROI. In this sample, I performed the region of interest scan in the center of the sample. However, the HeliScan can also perform off-axis region of interest scanning. Here we can see a 2D vertical slice from both the full field scan and the region of interest scan taken of the sweet gum seed. The two tomograms are closely aligned, but not perfectly aligned. So this red dotted box represents an area close to where the region of interest slice is taken. Let's zoom into these regions and have a closer look. We can see from these images that the region of interest scan has successfully allowed us to gain more information about the features in the matrix of the seed. So in summary, during this webinar, we've gone through an overview of the HeliScan and region of interest scanning, and have talked about how region of interest scanning is a good option if you want to obtain a tomogram with a smaller voxel size without having to physically cut your sample. If you're interested in learning more about the HeliScan and are interested in a couple of good papers to read, I would recommend the following two. This was our last Beyond the Scope webinar for this year. Our next webinar will be presented by Dr. Dan Vecti in January, and the date is still to be determined. Don't forget to stay connected with us via our website, LinkedIn, or Facebook pages. All the previous Beyond the Scope webinars have been uploaded to our YouTube page. Thank you for joining me today. I hope you all have a good evening. If you're in the US, if you're in Australia, have a good day. If you're in Europe, I'm not quite sure what the time zone is yet there. If you have any questions, please either use the raise your hand option now or add it to the Q&A box.